Good morning, class. Our today's topic is tissue system in plants. Tissue system in plants is totally based on the three types of the systems. In plant tissues, the outer covering tissues are known as the epidermal tissue system. In between, in the center position, the vascular tissue system that is composed of xylem and phloem. And around all these tissues that is known as the ground tissue levels or the fundamental tissue system. So plant tissue system is differentiated into the three parts, the three areas: epidermal tissue system, vascular tissue system and ground tissue system. Look at this picture. This picture shows the transverse section of leaf, stem and root. Leaf is going to be study the directions that is found in the upper picture side. The center position is known as the vascular bundle. Outer covering, brown coverings are the epidermal, and the green appearance is known as the ground tissue system. Similarly, in case of the stem, the brown surface area around this circle is the epidermal tissue system, the dermal tissue. Center position green color is the ground tissue, and in the center the purple color tissues you can find the vascular tissue system. So these are the basic arrangement of these three types of tissue system in leaf, stem and root. Epidermal tissue system is based of the formation of the outermost covering of plant body. It consists of epidermal cells, epidermal appendages and the outer surface structure that is known as the stomata, which is found on the outer surface of the leaf. Epidermis is the outermost layer and having always in the single layered structure. Each cell has a small amount of cytoplasm, but a very large size vacuoles is there. The outer surface is covered with the thick layer to lose the respiratory loss or the photosynthesis loss or heat loss. Sometimes the cuticles is present which prevent the loss of water from the aerial part of the plants. The epidermis is interrupted by the small pores that is called the stomata in case of the young stem as well as in the leaf upper and lower surface. This stomata is surrounded by the two guard cells. These are known as the guard cells is there. So stomata is responsible for the purpose of the transpiration. This is the cross section of a monocot root. In the monocot root, you can see here the outer area is the epidermis, center is the cortex, and after that a circle is just like an dermis area, that is endodermis. And just after the endodermis, a circle lining is known as the pericycle, and in the center you can find the pith region. The xylem and phloem tissues is seen in the vascular bundle formation and the outer surface area having a root hair region also. So this is the cross section of the monocot root. So the guard cells are the bean shapes in case of the dicot plants. Guard cells means the cells which is going to guard it, the stomata. In the dicot plant, it is bean shape or kidney shape, and dumbbell shape is found in the monocot plant. The outer wall is thin and inner wall is thick of these guard cells or stomata. They possess the chloroplast also and carry out the process of photosynthesis also. So the epidermal cells in their vicinity become the specialized in their size and shape also. And the stomatal aperture means stomatal aperture, guard cells and surrounding epidermal cells is there, is confined together to form the stomatal aperture or apparatus. Stomatal apparatus is there, and epidermis also bear the appendages sometimes. The root hairs are unicellular elongation of the epidermal cell in case of the root, and it is multicellular in case of the stem. So appendages on stem epidermis are called the trichomes. Trichomes are always in the multicellular structure. It helps in preventing the water loss by the process of transpiration, and granular or secretory cells is present in many plants, responsible for the secretion of certain chemicals, certain things in case of the plants, like resins and gums also. This is the typical structure, very typical structure, typical diagrammatic structure, transverse section. You can see here the primary xylem, primary phloem in the center area, and the root hair regions is also some root hair there. Cortex region is also there. The outer coverings are the epidermal or single layer cell, and then pericycle is also there, and vascular cambium and endodermis. You can see this is the typical structure of transverse section of any root hair, root zone, plant root system. In case of the vascular tissue systems, it is composed of the vascular tissues, means xylem and phloem. So this tissue system is known as the vascular tissue system. It includes the xylem and phloem. It occurs at a discrete strand that is called the vascular bundle also, means conjoined to form a bundle-like structure, and occur along some radius sometimes. Vascular bundle is called the collateral, means radius type of radius arrangement is known as the collateral type. When strips of the cambium tissue is present in between the xylem and phloem, the bundle are known as the open type of vascular bundle. But if there is no cambium in between the vascular bundle and in between the xylem and phloem, this type of vascular bundle is known as the closed type of vascular bundle, means presence and absence of the cambium tissue. You can also see here this with the help of this diagrams how we are going to cut the transverse section of the leaf, stem and root. Two phloem poles occurs in a vascular bundle that's separated from the central xylem. Then this condition is known as the bicollateral, or the vascular bundle are known as the bicollateral, means two phloem poles and in the vascular bundle that is separated by a central xylem, means xylem in the center and either side phloem tissues are present, bicollateral vascular bundle. In the radial vascular bundle, the xylem and phloem occurs along with the differentiated radii that alternating each other, means one xylem, then one phloem, then one xylem, then one phloem. When protoxylem is towards the periphery and metaxylem, means the mature xylem, the full size xylem is towards the center, the condition is exarch. And when protoxylem towards the center and metaxylem towards the periphery region, the condition of such type of bundle is known as the endarch condition. So there are the two conditions, exarch condition and endarch condition, exarch and endarch. This is the representation of a stem, a mature stem. You can see in this mature stem a secondary root. Two types of root is there, sap root, center position, and center is very hard root. So hard root and tap root, sap root, two types of root is there, in which you can see here the phloem fibers, xylem phloems, xylem vessels. Annual rings is there. Annual rings, it actually shows the annual rings means one ring is formed in a year. So number of annual rings is present in the plants is denotes that how much the age of that particular plant. So this is the sieve tubes is also there in the mature, and downwards and upward movements is showing the upward movements of water by in the vessels, and the downward movements by the sieve plates of the photosynthetes with the help of the companion cell also. Third type of the tissue system is found in the plant is the ground tissue system. Ground tissue systems means all tissue except the epidermis and vascular bundle. All tissues are known as the ground tissues. It consists of simple type of the permanent tissues, means parenchyma, that cell present in the cortex, pericycle, pith and medullary rays, and in the ground tissues in the leaves. The leaves are totally filled with the chlorophyll, that is known as the mesophyll cells. So mesophyll cells are responsible for the purpose of the photosynthesis in case of the ground tissue that is present in the leaf. You can see here the vascular tissue, ground tissue, and dermal tissue, xylem and phloem. A very typical arrangement of these tissues are shown by the transverse section if we are going to see this under the microscope. If we cut down the transverse section of the monocot root and compare it with the dicot root, we find this structure. In this picture, you can see the unicellular root hairs are found in the monocot root, but in the dicot root, the unicellular root hair is also there. In place of the epiblema, This is the epiblema. Starch granules is also there, but if you are going to differentiate these on the basis of the vascular bundle, you can see here the number of vascular bundles are very high in the monocot root in comparison to the dicot root. So on the basis of this vascular bundle, you can easily differentiate which picture is of the dicot root and which is of the monocot root. Similarly, if we are going to study the differentiation of a monocot stem and the dicot stem, you can easily diagnose this with the help of the microscope. The vascular bundles, if found scattered in a bundle stage in all the ground tissues and no root multicellular stem here is there, then it is the monocot stem. And in case of the dicot stem, it is found in the ray-like structure, that is the medullary ray-like structure. So if medullary rays is there and the bundles in form of the metaxylem and protoxylem, xylem parenchyma is also there, the pith is there in the center, it is known as the dicot stem. So on the basis of the vascular bundle, you can easily differentiate the structure or the transverse section of the monocot stem and compare it with the dicot stem. Similarly, in the dicot leaf and monocot leaf, you find there on the basis of the stomata there, the vascular bundle is also there. The vascular bundles is found the stomata in the center, and the stroma or the on the lower surface of in case of the dicot leaf, and in the isobilateral leaf or in the monocot leaf, it is found in the upper and lower both the surface. So the dorsal leaf is known as the dorsiventral leaf and monocot leaf is known as the isobilateral leaf. So in case of the isobilateral leaf, you can easily see the mesophyll cells is there filled of the chlorophyll, and in the substomatal cavity there in centers, the xylem and phloem are in form of the bundle tissues in a very large amount. So parasite mesophyll parenchyma is found in the dicot leaf, and parasite parenchyma mesophylls is there not present if present in a very short forms. So it is known as the adaxial epidermis also. So mesophylls tissues are responsible for the purpose of the photosynthesis. The air cavity is found in the dicot leaf and spongy mesocell cells is there, and no air cavity or sometimes very few air cavities found in the isobilated or in the monocot leaf. So you can differentiate the dicot leaf and monocot leaf with the help of the permanent slide through the high power microscope also. This is all for the total study of the tissue system in case of the plant. Thank you and have a nice day.